Science just broke the rules of reproduction, and the world may never be the same. What if science could push the boundaries of nature itself? That's exactly what a groundbreaking discovery has done. Scientists have created a mouse with two biological fathers. Could it challenge the very definition of parenthood? So stick around till the end to watch how this incredible feat could change the way. But before, don't forget to hit the like button and subscribe to our channel to watch more mind-blowing scientific discoveries. The virgin birth, parthenogenesis. In nature, the concept of a virgin birth, where an organism is born without a father, is surprisingly common among certain species. This phenomenon, known as parthenogenesis, occurs in some animals when mates are unavailable or when such reproduction offers a survival advantage. Essentially, an egg has all the necessary components to develop into an organism without fertilization. But could this natural occurrence inspire breakthroughs in the lab? For most animals, though, parthenogenesis isn't the preferred method of reproduction. It's usually a rare occurrence rather than the norm. However, scientists were inspired by this natural process and wondered if they could manipulate it in a laboratory setting, particularly in mammals. In some cases, this unique form of reproduction might even serve as a solution to challenges in animal conservation. The possibility of asexual reproduction could allow endangered species to reproduce even without the presence of males, sparking excitement in the scientific community. The first experiment using two mothers. In 2004, Japanese researchers attempted a bold experiment. They tried to create a mouse using the genetic material of two mothers with no father involved. This led to the birth of a mouse named Kaguya, the first mammal successfully created with two mothers. What challenges did this pioneering experiment bring to light? To achieve this, the team genetically modified one of the mother's eggs to behave like sperm. By transferring the nucleus from this modified egg into the egg of the other mother, they created a viable embryo. While Kaguya's birth was a significant breakthrough, the process wasn't flawless. Many embryos didn't survive, and only a few mice reached adulthood. Yet Kaguya's birth sparked curiosity about the implications for reproductive medicine, as it introduced the concept of creating life without male involvement. It also raised questions about the viability of such experiments in the future. This experiment highlighted how much we still need to learn about embryonic development, but it also opened the door to further innovations in genetic research. Despite the excitement, scientists were cautious in their optimism as the success creation of father's eggs and first failures. In March 2023, a major milestone was achieved. Chinese researchers created eggs from the cells of male mice and successfully fertilized them with sperm from another male mouse. This resulted in the birth of 7 live mice, all with two biological fathers. What did this experiment reveal about the potential of male-based reproduction? However, the process was far from efficient. Out of 630 embryos created and implanted into surrogate mothers, only 7 mice survived. This low success rate suggested there were still many unknown factors at play. One challenge was the need to grow the stem cells in a controlled environment, where they could lose their Y chromosome and become suitable for egg formation. The eggs were then treated with a chemical called reversine, which helped promote their development but might also have caused genetic damage. The experiment underscored the fragility of this kind of scientific progress and served as a reminder of the limits of current genetic technology. Chinese experiment that did it again, but better. In a follow-up study, Chinese scientists improved the process. This time, they used advanced genetic editing to address a critical problem: genomic imprinting. This issue occurs when genes from both parents are expressed incorrectly, leading to developmental abnormalities. Could this new breakthrough pave the way for healthier and more viable offspring? Using precise genetic techniques, researchers corrected 20 different imprinting issues in the embryos. As a result, the survival rate increased from 1.3% in earlier experiments to 13%. The mice born in this study were fully developed and healthy. However, there was a significant drawback. These mice were sterile, meaning they couldn't reproduce. This raised a major question. While these advancements in genetic engineering were remarkable, would the ability to create life with such precision come with unintended consequences? Mammalian barrier to unisexual reproduction. While unisexual reproduction, using only one sex for genetic material, is common in some species, mammals face significant genetic barriers that make it difficult. These barriers likely evolved to ensure genetic diversity, which is essential for the survival of mammalian species. Are these genetic roadblocks insurmountable, or can they be overcome with further research? One major obstacle is genomic imprinting. When an embryo inherits DNA from two mothers or two fathers, it can result in imbalances in gene expression. These imbalances can cause severe developmental issues or prevent the embryo from surviving. Despite these challenges, scientists are finding ways to overcome some of these barriers, as demonstrated by the recent success in creating mice with two fathers. Each breakthrough in overcoming these barriers brings us closer to unlocking the secrets of reproductive biology, offering hope for addressing infertility and other reproductive challenges in the future. Additional problems and why even do this. While these experiments are scientifically impressive, they also raise questions about their purpose and practical applications. Why are scientists pursuing this line of research? There are several reasons. What drives these researchers to push the boundaries of genetics so far? These studies help us learn more about the fundamental mechanisms of reproduction and embryonic development. This understanding could eventually help tackle infertility issues, providing new solutions for couples who struggle to conceive. The techniques developed in these experiments could be applied to other areas of genetics, such as treating infertility or preventing genetic disorders. Additionally, the ability to manipulate DNA in this way may lead to breakthroughs in gene therapy, ultimately allowing scientists to correct genetic conditions at an earlier stage of development. Exploring these possibilities pushes the boundaries of science, but it also raises ethical questions about the implications of creating life in unconventional ways. As these experiments progress, society must grapple with the moral dilemmas they present. While creating human embryos with two fathers or two mothers is not currently feasible or ethical, the knowledge gained from these studies could one day contribute to advancements in human reproductive medicine. The potential applications could extend beyond fertility, influencing genetic engineering and even the prevention of genetic diseases before birth. The creation of mice with two biological fathers represents a remarkable scientific achievement. It demonstrates the potential of stem cell and genetic research to address fundamental questions about life and reproduction. While there are still many hurdles to overcome, these studies pave the way for future discoveries that could revolutionize reproductive science. As we continue to unlock the secrets of genetics, the future of reproduction could be forever changed. As these frontiers of reproductive science evolve, the question remains, how far are we willing to go in the pursuit of life and innovation? So, what do you think about this incredible scientific breakthrough? Could this spark a new era in genetics and reproduction? Share your thoughts in the comments below. And remember, if you want to stay up to date on more cutting-edge discoveries like this, make sure to like, share, and subscribe to the channel. The future is unfolding right before our eyes, and with the right knowledge, we can shape it. Thanks for watching. Stay curious, stay informed, and we'll see you in the next video.